Good morning to everyone. Thank you for coming to this session. I would like, first of all, to thank the ALPS committee to give me the opportunity to give this talk and for inviting me. It's been already pretty remarkable, and thank you so much. Anyway, so I will try to, in this talk, to overview basically what is the mechanism of action of psychedelics compound, with particular emphasis in LSD and the ability of LSD to modulate social behavior, alcohol use disorder, and anxiety. I'm very affectionate to this molecule because I started to work with this molecule since so far almost 10 years now. So those are my conflict of interest. And what I would like to point out is also to dissect as a basic pharmacological guy and scientist, what we do in our lab. We try to dissect what are the brain regions involved in the potential therapeutic effects of those compounds, and what are the neurotransmitters involved. So first of all, as many of you already know, but I like to always point out this, that, hallucinogenic compounds, usually they include different, we can divide the hallucinogenic compounds in two main classes: psychedelics or social classical serotonergic hallucinogens such as psilocybin, LSD or mescaline, and dissociative anesthetics such as ketamine or phencyclidine. But psychedelics are also called classical serotonergic hallucinogens, not only because, as you know, they act as partial agonists of the 5-HT2A receptor, but also because they share with the serotonin the similar chemical structure, and we do know that serotonin is a neurotransmitter particularly important for the regulation of mood, sleep, and anxiety. But why LSD is so particular? Because LSD is a drug that we can define pretty promiscuous, because does not only binds the serotonin 5-HT2 receptor, but it's a drug that works with a very pleiotropic mechanism of action involving 5-HT2 receptor, dopaminergic D2 receptor, trace amino associated receptor 1 in different brain regions. So it's not only about the middle prefrontal cortex, but also the brain stem, such as dorsal raphe nucleus, which is rich of serotonergic neurons, ventral tegmental area, and the activation also of the 5-HT2 receptor is correlated to an over-release and spillover of glutamate, which in turn activates other receptors, such as NMDA, but also AMPA. We got pretty fascinated by this molecule because, when I started to work on this molecule, it was 2014. I was doing my postdoc in Canada, and we heard about this echo concerning the fact that different professionals and entrepreneurs in the Silicon Valley were using to take low doses of psychedelics, particularly LSD, to boost their creativity, to boost their empathy, and social behavior. And since then, many papers started to come out in order to highlight the potential ability of those compounds to enhance social behavior. And we mostly hear about this concept, microdoses. And here in Switzerland, of course, there is Professor Liechti, who strongly studies, the impact of low and higher doses of LSD to modulate social behavior. But how can be defined social behavior? Social behavior is something which is very shared by all the mammals and all the individuals, and it can be defined as a plethora of different interactions among individuals, and dysfunction of social behavior are common in different psychiatric disorders as autism spectrum disorder, social phobia, and major depressive disorder. So we got very fascinated, by those human studies, and as neuroscientists and basic psychopharmacologists, what we do in our lab is a back-translating approach. So we start from the human data and we try to back-translate in animals in order to understand what are the neuronal circuits involved in this mechanism of action. So for this reason, we published a few years ago an article in which we demonstrated that low non-hallucinogenic doses of LSD in rodents enhanced social interaction. So we performed this test, which is called direct social interaction, in which tested mice were treated for 7 days with low non-hallucinogenic doses for mice, which correspond to 30 micrograms per kilos for 7 days, and we saw that in those tests, animals that were treated with LSD were interacting more, And we demonstrated that by pharmacologically blocking 2 receptors directly in the middle prefrontal cortex, and those receptors were the 5-HT2A and the AMPA receptor. So we microinfused in the medial prefrontal cortex of those animals a selective antagonist of those 2 compounds, of those 2 receptors, and the prosocial effect was completely blocked and nullified. Of course, we try to go a little bit more in the details about the potential molecular effect of those drugs. And I mentioned the AMPA receptor. It is very well known that the activation of AMPA receptor leads to the molecular potentiation of a particular molecular signaling, which is the mTOR, mammalian target of rapamycin, has been demonstrated by the group of Ronald Newman and John Crystal and Yale University. And this mammalian target of rapamycin is strongly important for the neurobiology of different mood disorder, anxiety disorder, but also schizophrenia, drug addiction. And the mTOR is basically characterized by 2 macrocomplexes of protein, the mTORC1 and the mTORC2, and in turn, each complex is characterized by many other tiny proteins which are all interconnected each other. And in our animals, we found that in the middle prefrontal cortex, the LSD administration increased the levels of the phosphorylated level of the phospho-mTOR in the middle prefrontal cortex, and also another protein, the phospho-AKT, which activates in turn mTOR. So we questioned basically how much this complex of proteins is important for the prosocial effect of LSD. So we generated a line of genetic mutant mice that were lacking a structural component of the mTOR, the raptor, selectively in the glutamatergic neurons in the middle prefrontal cortex. And basically in those animals, here in gray, you can see the wild-type animals, which means animals that did not undergo any genetic mutation, and the LSD was able to exert the prosocial effect. So the interaction time in their tiny little cage was higher. But in those mice, there was no difference between, vehicle and LSD, so it means that in those mutant mice lacking this structural component selectively in the glutamatergic interneurons is strongly important. Then we did another experiment in another strain of genetic mutant mice that were basically, they didn't present this protein in the GABAergic interneurons, but here LSD was pretty working. So LSD is important for modulation of social behavior through the activation of the mammalian target of rapamycin selectively in the glutamatergic neurons. But cortex is essential, for many biological functions and also for the modulation of social behavior. Indeed, damage of the human prefrontal cortex has been associated with impairment of the social behavior. So we performed another experiment, which is called the optogenetic approach. So with the expression of a specific virus in the brain, we were able to selectively shut down the activity of the pyramidal neurons, glutamatergic neurons, in the middle prefrontal cortex. And basically what we found is that when the activity, the neuronal activity of those pyramidal neurons, glutamatergic neurons, in the middle prefrontal cortex was shut down, LSD was not able to exert the prosocial effect. So this means that the prosocial effect of LSD is primarily mediated by the 5-HT2A receptor and AMPA receptor for sure, and that the integrity of the middle prefrontal cortex is fundamental for the ability of LSD to produce social behavior. Then, of course, different studies, many studies start to come out highlighting the ability of LSD to exert anxiolytic effect. Anxiety is a multifactorial disorder which is characterized by both physical and psychological symptoms, and of course can be caused by drugs, life events, genetic factors that can play a crucial role in the development and in the neurobiology, basically, of anxiety. So when we talk about anxiety, what is the neurotransmitters that is strongly involved in the neurobiology of anxiety disorder? Well, it's not the only one, but surely serotonin plays an important role, not only for anxiety disorder, but also for major depressive disorder. And this is pretty known, that dysregulation of the serotonergic transmission is strongly involved in the neurobiology of anxiety. And when we talk about serotonergic transmission in the brain region that contains the highest amount of the serotonergic neurons is the dorsal raphe nucleus, which is a brain region that connects, projects its inputs to the middle prefrontal cortex, and which is very and extremely sensitive to condition of stress. So in this case, in the same way, we developed this back translational approach. So we saw those human data that were published, so we tried to understand, okay, but what is the mechanism of action, the anxiolytic effect of LSD? In 2022, we published this paper in which we investigated the serotonergic neurotransmission in a rodent model of anxiety disorders and the impact of the same dose regimen of LSD that we saw to enhance, to boost social behavior. So, of course, we needed to make animals anxious, and to do so, I don't know if you're familiar with behavioral tests, but basically we use this approved protocol, which is called chronic restrained stress, for 2 hours per day for 15 days. And we wanted to see, for now, if so the chronic stress lasts 15 days, and we administered LSD for 7 days in the last 7 days of the chronic stress procedure, and then we performed electrophysiological activity recordings and behavioral tests. But first of all, we wanted to understand what is the effect of LSD in non-stressed animals on the serotonergic activity. So we perform an electrophysiological analysis and neuronal recordings of the serotonergic neurons in the dorsal raphe nucleus. So what happens when we acutely inject LSD at 5, 15, and 30 microgram per kilos, which are in animals non-hallucinogenic dose? We see basically a shutdown, a reduction of this activity. But what happens when we administer in those animals LSD for 7 days? There is a switch. So there is an enhancement of the neuronal activity and also a change in some electrophysiological patterns which are mediated by another receptor, which is the serotonin 5-HT2A receptor. And how we found this? Because with a technique which is called microiontophoresis, we're able to selectively modulate single receptor in that specific brain regions. So what happens when we microiontophoretic inject this drug, which is pretty difficult to pronounce, H-OIDPAT, which is a selective agonist of this 5-HT2A receptor. So in animals treated with vehicle, when this receptor is activated, we see basically a reduction of the neuronal activity. Why this? Because the 5-HT2A is a presynaptic receptor. So in pharmacology, when you activate a presynaptic receptor, you have less release of the neurotransmitter in the postsynaptic cleft, which is translated in a reduction of the serotonin at the postsynaptic transmission, and therefore a reduction of the neuronal activity, so the firing rate. This didn't occur in animals that were treated with LSD. So it means that the 5-HT2A receptor is desensitized, so it's not able to transduce the signal. And if we think about, this is not very different to what happened to many drugs that are used to treat anxiety, but also major depressive disorder. For example, SSRI drugs, the selective serotonin reuptake inhibitors. Because at the beginning of the treatment, it has been demonstrated that when drugs like citalopram or escitalopram are administered, we have a decreasing of the neuronal activity. But after long-term administration, we have a readout on the firing rate. So we have an enhancement of the neuronal activity, and that is why patients, when started treatment with selective serotonin reuptake inhibitors, at the beginning of their treatment, they feel even worse because there is a shutdown on their initial firing rate activity of the serotonergic neurons, which doesn't occur when the treatment is chronic. And what happens when you have anxiety disorder in the dorsal raphe nucleus at the serotonergic neurons? So animals following chronic stress procedure, they have, as you can see, a reduction in the neuronal activity. And we found that the pretreatment with LSD was able to prevent this decline in the neuronal firing rate. We coupled those electrophysiological experiments with some behavioral tests because, of course, you are not able to ask to the animals how they're feeling. So you perform some behavioral tasks, and one of these, I will not go into so much in the details. We perform different behavioral tests, but this is just the most clear. You put basically the animals, to assess anxiety in this open field arena, and what you measure is the time and the number of transitioning in the center of this arena, because an arena is extremely anxiogenic for an animal. So the animal with anxiety, they try to stay more in the perimeter. And what we found, of course, that animals that were stressed, of course, they don't want to stay in this center, they're pretty scared, they stay mostly in the perimeter, and LSD is slightly normalized. So we perform also other behavioral tests, but we don't have so much time, so I just want to show you those data. And we found also that stress is correlated to a reduction in the neuronal plasticity, and stress and depression, stress leads to depression and anxiety. And this is very well demonstrated that when we analyze the pyramidal neurons in the middle prefrontal cortex, there is a reduction in the number of spines. You know, the spines are able to connect each neurons to the others, and chronic stress produce a reduction of the synaptic buttons in the pyramidal neurons, and LSD already in the control animals was able to enhance the number of the dendritic spines, and of course was able to prevent the reduction of those spines that we're seeing in the chronic stress mode. So the neurobiological and pharmacological mechanism to LSD to prevent anxiety is occurred through the mediation and the modulation of the serotonergic transmission. At least that was the area that we investigated, but LSD is a molecule that acts in different brain regions with a very pleiotropic mechanism of action. Until now, we talked about low doses, low non-hallucinogenic dose. But what happens when we administrate a proper hallucinogenic dose in animals, but not only in humans? So a full hallucinogenic doses in humans, but this is not the only one, even lower than this, of course, produce some symptoms that are extremely comparable with what happens occur in psychosis or schizophrenia. You know, we can see body distortion, delusion, euphoria, dysphoria, depression, interoception, working memory impairment, of course, and even in animal models, higher doses of these compounds reduce social behavior, induce aggressive behavior, and those animals basically at least they experience a so-called transient psychosis that can last 24 hours. And what we tried to demonstrate a few years ago is that apparently this modulation, these transient psychotic behavior are due to the modulation of the dopaminergic neurons in the ventral tegmental area, which is a brain region strongly involved in the neurobiology of psychosis and schizophrenia, because as we can see, the higher doses of animals can produce a shutdown of the neuronal activity of the dopaminergic neurons into the ventral tegmental area. But it's not only about VTA, because an important circuit, the thalamocortical circuits, represents one of the potential neuronal circuits of consciousness. And these functions of these circuits have been strongly correlated on the development of different diseases such as psychosis, schizophrenia, but also autism spectrum disorders. These circuits is also called the doors of consciousness, and it is characterized mostly by three nucleus: medial dorsal thalamus that projects glutamatergic impulse to the reticular thalamus and to the cerebral cortex. And as you know, different clinical studies have been demonstrated that higher doses, human imaging studies have shown that psychedelics compound inducing LSD are able to strongly impact this thalamocortical circuit and eliciting a state of increased brain entropy, you know. That includes also, which is accompanied by thinking expansion, depersonalization, thus representing an important tool also to understand also how brain circuitries are connected each other, you know. And I don't want to go too much into the details, but what we published a paper a few years ago in which we wanted to investigate how those higher doses of LSD are selectively able to impact all those brain regions and particularly all those neurotransmission. In order to get this talk less boring, I just put these slides to summarize our finding. So higher doses of LSD, I'm talking about 150 micrograms, acutely, enhanced the neuronal activity of glutamatergic neurons in the medial prefrontal cortex, enhanced the neuronal activity of GABAergic interneurons in the mediodorsal thalamus, and also the reticular thalamus was particularly interesting because we found two different kinds of subpopulation of GABAergic interneurons. Some neurons responded to an enhancement of the neuronal activity, other neurons responded to a decreasing. We didn't go through yet why those different subpopulations responded in a different way. Probably it will be one of the next steps. But again, until now we talked about anxiety, social behavior, but also when I relocated back to Italy and I opened my own lab, I was looking at the literature and I started to get also pretty intrigued by another disease, which is alcohol use disorder. Alcohol use disorder is considered a major burden for the society due to the immense economic cost and the impact on, cognition, because we know that ethanol is strongly able to modulate and impair learning and memory process. Ethanol is able to produce very well-known locomotor effects, but also it is a chronic ethanol exposure. It's able to induce the development of cardiovascular disease, but also neuropsychiatric disorder. And we have drugs that are able to treat alcohol use disorder, disulfiram, acamprosate, and naltrexone. But those drugs are characterized by different side effects, and so in light of this, current treatment of the alcohol use disorder are limited and new therapeutic approaches are definitely needed. So as I'm a circuit guy, when we talk about alcohol use disorder, what are the brain regions that are interconnected to each other and modulate the neurophysiology and the neurobiology of alcohol use disorder? So for sure, the ventral tegmental area that projects the dopaminergic inputs to the nervous accumbens, which in turn produce projections to the medial prefrontal cortex. But also we do have some direct dopaminergic projections from the ventral tegmental area to the prefrontal cortex. And again, in this case, we started from clinical data. We started to see that different meta-analyses highlighted the potential use of psychedelics, particularly LSD, to treat alcohol use disorder. So we were looking at the mechanism of action, and we started to outline a potential study. We do know that LSD works not only as a 5-HT2A agonist, not only as a 5-HT1A agonist, and not only indirectly activates the AMPA receptor, but LSD works also as a partial agonist of the dopaminergic T2 receptor. This receptor is strongly involved in the modulation of the neurobiology of alcohol, because it has been demonstrated that chronic alcohol consumption is able to enhance the neuronal activity of the dopaminergic neurons in the ventral tegmental area through a desensitization of the 5-HT2 receptor. Whereas we demonstrated that LSD is able to shut down the neuronal activity of the dopaminergic neurons through the modulation of the dopamine 2 receptor, because if you block the dopamine T2 receptor, you don't have this modulating activity of LSD. So we decided to embrace this new path and try to investigate the mechanism of action of LSD and its anti-addictive properties. And of course, to do that, we use mice. But mice, usually they don't like alcohol. They don't drink alcohol, so you need to get used the animals to drink. And to do so, we employ this paradigm, which is called the drinking in the dark, which is last. 6 weeks, and the mice were subjected for, each cycle is characterized by 7 days. So the first 4 days of the week, mice had access to 1 bottle of water and 1 bottle containing the 20% of ethanol. The other 3 days of the week, so basically long weekend, no alcohol for them. Then, of course, we treated those animals with higher doses of LSD, which is acutely is able to produce hallucinogenic effect. But what happened in long term? LSD was administered alone or in combination with the 2 selective antagonists, one of the 5-HT2A receptor, ketanserin, and one of the dopaminergic T2 receptor, haloperidol. Many of you are psychiatrists, so you know that haloperidol is also used to treat schizophrenia. And then we monitored the alcohol consumptions for 40 days, and we measured the consumptions of alcohol during all those 40 days. We performed also some behavioral tasks. We assessed the locomotor effect, and also we measured the neuronal electrical activity of the dopaminergic neurons in the ventral tegmental area. So, thanks to my former postdoc, Carolina, who spent weeks and weeks in the lab trying to collect those data, what we found that 1 single dose of LSD, 150 micrograms, reduced the alcohol consumptions after 40 days, compared to vehicle. And when we compare those data with the animals treated with the selective antagonists of either 5-HT2A and dopamine T2 receptor, we found that the group that was pre-treated with haloperidol was not drinking. So it seems that in this test, the alcohol consumption is mostly mediated by the dopaminergic T2 receptor. Because if we see at the group that was in red, the group that is pre-treated with the ketanserin, they're still drinking. So in this test, the dopaminergic T2 receptor has a particularly important relevance. So, of course, we know that alcohol consumption is able to induce locomotor impairment. So we perform 2 tasks. This task we already saw previously to test anxiety. And animals, the open field test, so there was no effect on the locomotor activity and no effect on the number of entries into the center. So in this case, no locomotor impairments were detected. But what we found is that when we measured another performance in the locomotor assessment, which is the rotarod test, so basically the rotarod is a machine which is rotating, and when we put the animals, of course we measure the latency to fall, and which is a sign. As earlier the mice fall down, this is a sign basically of low performance, so they have some impairment. And compared to the animals that they never drank, of course, mice treated with chronic alcohol consumptions, they fall down immediately in less than 50 seconds. And this effect was totally normalized by the LSD after 40 days. So we don't see any more locomotor impairments. We need to increase the number of animals, but it seems that both haloperidol and ketanserin slightly block this effect. There is a trend in this case, but we need to increase the number of animals. Then we decided to perform electrophysiological recordings of the dopaminergic neurons in the ventral tegmental area. So what happens in animals following chronic alcohol consumptions? We have, compared to the naive animals that were they were not drinking, we have an enhancement of the neuronal activity of dopaminergic neurons, and LSD totally normalized. It seems that at least for the neuronal activity, either the T2 or either the ketanserin receptor were not involved. there is something more complicated going on. And we measure also another parameter, which is called the burst activity. So what is the burst? The burst in the electrophysiology is a particular event which is characterized by an over-release of neurotransmitters in a specific range of time. Each neuron has its own range of time. For example, dopaminergic neurons has a particular range in which those different action potential spikes. Serotonergic neurons, other parameters, glutamatergic neurons, other parameters. And what we found is that correlated with the higher activity, those neurons firing more in bursts. So we have an over-release of dopamine, which is nullified and normalized by the chronic, by the single administration of 150 microgram per kilos of LSD. So what is intriguing is the fact that LSD is able to produce long-lasting effect, because what we are seeing here is measured 40 or 45 days after the single administration. And there are some hypotheses, and we are performing some DNA methylation experiments to understand what are the epigenetic changes that LSD is able to perform in those brain regions. So what we can say is that, for this third part of the talk, is that single injection of LSD reduced alcohol consumptions primarily through the dopamine T2 receptor in a preclinical model of alcohol use disorder, and LSD also partially restores the dopaminergic alterations induced by chronic alcohol consumptions. Concerning the electrophysiological effect, the story is even more complicated, because apparently those receptors, dopamine T2 and 5-HT2A, seems to be not involved. We have some hypotheses, but we need to test. So I basically, something that I didn't mention you, so some of you might thinking why you didn't try the low doses that you showed us before. We tried, but the low doses of 30 micrograms per kilos for 7 days that were able to induce prosocial effect and also anxiolytic effect, they were not exerting any kind of effect in this model. So for alcohol use disorder, we needed to inject at very high doses to produce those long-lasting effects. Where is going now the current line of research? So LSD is a molecule which is very promiscuous. It works with a very different pleiotropic mechanism of action. What we know so far until now is that hallucinogenic compound and the research is trying to synthesize compounds that are more selective and that are not able to produce visual hallucinating effect. So there were some studies that have been published in which selective compounds that might be not induce hallucinogenic effect might be characterized by a very selective mechanism of action that involves only one receptor. Nevertheless, I'd like to conclude all my presentation by saying that, as Professor David Nutt already overviewed in 2007, that despite everything, psychedelics are still the least dangerous mind-altering substance. If you can see in comparison with the alcohol, you can see that LSD and mushrooms are quite on the bottom of this, So I would like to thank you so much. Particularly, I take the occasion to introduce the Italian Society of Psychedelic Medicine, which has been founded just very recently. And I'm part of the scientific board, along also with Federico Seragnoli, and Tommaso Barba, who works with David Herzog. And this is the directive teams and founders. Thank you so much for your attention. Thank you so much for also to my team in the San Raffaele University in Milan. This is our campus funding agency. And thank you so much.
Thank you so much.
Try to stick with the time in order to give people time to go to the coffee break. So.
Yeah, we have more time than this.
Okay.
Yeah, don't worry. So a couple questions.
Hello, thanks for your very interesting presentation. So I have a little question. It's more general, but since you have been working on LSD, it's something. It has always been a question to me. Actually, it's about the dose of LSD. In the very fact that it's micrograms, and then it enters the bloodstream and it goes to the brain. But what I don't understand well is how, because it's micrograms, how it can influence so much surface of the brain.
Well, it's a very good question for a pharmacologist. You know, everything depends on what is called the binding affinity. So no matter how much is the dose, but what is important, what we call two very important factors. One is the binding affinity, which is the ability of the drug to interact with the receptor. But not only that, the real question is how much the drug is able to activate the receptor, which is parameter which is called intrinsic activity. So no matter how much the dose is, the intrinsic activity of the LSD to activate all those receptors that I mentioned, particularly 5-HT2A, of course, dopamine T2 receptor, 5-HT2A receptor, you need just a very low amount. And it is not only this the matter. The thing is that it depends also by the concentration of the receptor in that specific brain region. So the middle prefrontal cortex is constellated by the 5-HT2 receptor. And this can justify the level of occupancy of the LSD. If you read some brain imaging studies and if you see the level of occupancy of the LSD in those brain regions and in those receptors, you will realize that it doesn't matter how much low is the dose. But those molecules, you just need a really tiny amount to affect the behavior. Even though those drugs, the half-life of those drugs might be very quick, because LSD stays in the body for 12 hours, But still, this drug is able to produce long-lasting effects that we are not understanding. I mean, we can detect in animals, but we were having a conversation, last night, and in humans it's still not very easy to understand this pleistogenic ability of those compounds, because in animals we can measure the number of dendritic spines. In humans, we're still in vivo. It's not so obvious, We can measure the plasmatic level of BDNF, but this can say everything and nothing, It's just a correlation.
Thank you.
Here.
Yeah.
Congratulations, this is really hard work. I've been in the rat mice electrophysiology myself in the past. But I have a question that we also know that these animal models for drug addiction, abuse, and psychiatric drug dependence do not translate very well. The most famous case being the rat park study in Canada. And I wanted to hear your opinion on what can we actually reason in translating from these animal models to actually trying to treat drug addiction or dependence with LSD and psychedelics?
Yeah, so this is a very important question. I mean, this is not only a problem of drug addiction, but this is also a problem also for other psychiatric disease models, such as major depressive disorder or anxiety. What we can appreciate is mostly, we can appreciate a readout of this behavioral effect, but what we try to understand is the neurobiological, physiological mechanism. I mean, then, for example, here in the alcohol use disorder models, what we found, for example, is the ability of LSD to reduce the consumption, but we didn't test and we don't know what happens during the withdrawal syndromes, also. And what we can assess for surely is the neurophysiological impact. There are some tests which are controversial. Even the test that allow us to dissect, the major depressive disorder. There are some researchers who start to question it, how much we can predict, for example, the relevance or the face validity of the four-swimming test, which is used to assess major depressive disorder. But until now, those screening tests are helping us to, to discover a huge amount of molecules that are used to treat patients. Of course, what we can extrapolate are not all the core features, but for sure, the molecular biological mechanism of action surely is something that we can extrapolate, with all the limitation that can come out, Many hands raised. I don't know where to start.
Oh, excuse me. Yes, apomorphine, a dopamine D2, D3 agonist, has a long history as a therapeutic agent for alcohol abuse disorder, actually more than 100 years, although it's never taken off clinically. So, and there are rat studies showing that D2 agonists attenuate alcohol consumption acutely. But do you think that there's something about LSD's interaction with the D2 or D3 receptors that accounts for the long-lasting effects? Could something about its kinetics leave an imprint in some pathway?
Yeah, I mean, actually, there was in my slides, there was, after the LSD, when we saw that the LSD produces a shutdown of the neural activity, we inject apomorphine because we know that apomorphine selectively act on the dopaminergic neurons to just confirm that the neurons, was dopaminergic. So that drug showed up in my slide. But to answer to this question is very challenging because we know that from a behavioral readout, when we block the dopaminergic receptor, those animals, they drink less. But we don't see this effect in the electrophysiological activity. One of the hypotheses is that dopaminergic neurons and VTA express also 5-HT2C receptor, which colocalize with the dopaminergic receptor. So one possible answer is that, and we know also that LSD activates the 5-HT2C receptor. So one of the hypotheses is that maybe there is a crosstalk interaction between those two receptors which might colocalize and enhance this and produce this therapeutic effect, or maybe not. Or maybe, as someone suggested, you need the activity of all those receptors to work correctly in the same time. So maybe an experiment would be to use genetic mutant mice that they don't present both 5-HT2A dopamine receptor and or eventually 5-HT2C receptor, and we might see something, but I don't know the answer for now. In front here. Oh, maybe, yeah.
Thank you. First of all, thank you for your great talk. I have a question, and unfortunately I missed the first minutes of your presentation. That's why I don't know if you answered my question or not. Is there a difference in terms of effect if I take it orally or via injection? Thank you.
Yeah, well, the main difference is the bioavailability, because if you take the drugs orally, of course, the drug needs to pass through the gastrointestinal tract, and before going to the bloodstreams, it takes longer, So after taking orally, the drugs has an onset of 30 minutes before to exert the acute effect. When you inject intravenously, when you inject the drug intravenously, there's no absorption because you directly inject the drug into the bloodstream. So the effect comes out in few seconds. So that's the main difference, about the onset of the behavioral effect. Yes, front here.
Magnificent talk, thank you very much. From the first half, schematically, I understood that microdosing of LSD through a different mechanism increases serotonin in the synaptic cleft. Are there studies comparing SSRIs and microdosing of LSD?
Yeah, there was a study which has been performed by the Professor Blier in University of Ottawa in Canada, he's a psychiatrist, but he also works on animal models, and he likes to perform those comparison studies between SSRI and psychedelics. And in some experiments, depending also what strain of animals you use, because depending also on the distribution of the receptor, But in most of the case, the ability of LSD to enhance the serotonergic activity is superior to comparison to the escitalopram or citalopram, also because citalopram and escitalopram takes at least 2 weeks to enhance the electrical activity, even 3 weeks. LSD was able to enhance the activity just after 1 week. Still, it has to be understood, but probably it depends on the ability of LSD to activate the 5-HT2A receptor, which is responsible through the desensitization of the internal downregulation to produce the spillover of serotonin on the postsynaptic cleft,
I have one more question. Is there a possibility now to do double-blind randomized tests in humans comparing these two techniques?
Well, the problem is that we cannot selectively measure the electrical activity of a specific neurotransmitters. In humans, what we can perform is an EEG, but with the EEG, you can measure a power spectrum of a general neuronal activity. You put some electrodes in the where the hippocampus is or the prefrontal cortex, but you cannot appreciate selectively the real-time electrical activity of a specific neuron. So in humans, you could do, but it could be very invasive. So. The only thing that you can do, you can measure the receptor occupancy by using PET imaging studies, but still, sometimes it's not a direct reflection of what is the neuronal activity. Of course, there are studies that have been published comparing the effect of escitalopram with psilocybin. There are all the studies of Robin Cartaris, but it's not the direct studies concerning the electrical activity of the serotonin neuron. Also because I just talk about serotonin, but might be not the only one neurotransmitters which is involved,
I just wanted to acknowledge that we have an altar outside in the foyer regarding animal testing and psychedelic science. And I haven't heard any acknowledgement for your subjects or any empathy for their contribution in your research. So I would just like to point that out.
Yeah. I mean, all our behavioral tests have been performed according, all the ethical and animal protocols, that been, so everything that we do is selectively approved, and each behavioral test needs 1 year at least of waiting for the approval from each Ministry of Health, so everything that we perform is performed through all those ethical,
I myself am very thankful to the mice who gave their life for your research, so just wanted to have a moment of consciousness for them.
Thank you.
We have time for one more question.
Thank you. I was wondering, was there a control group which received just haloperidol and whether it's possible that that played a role in impairing the addictive processes and reducing the amount of drinking.
No. I didn't show the data because, but we did add another batch of experiment by injecting all the antagonists. You have all to try to test all the antagonists alone to see if there is any effect in long term. We tried also single, repeated, no effect of, at least at the dose that we picked up. And the dose that we chose, they are enough to block selectively that receptor, but on their own, there was no effect on either locomotion or either electrical neuronal activity.
Okay, thank you.
You're welcome.